now. So we are on record, and we will post this to KBS's website later. Thanks. Go for it, Jim.
Yeah, I'm very happy to introduce Laura Podzikowski, who is a PhD student in EEB in her 4th year, I think. And she's going to be presenting on plant biodiversity effects on productivity and ecosystem functions. So, Laura?
Okay. Oh, intriguing. One moment. That was a little spoiler. All right. Okay, so biodiversity loss on a global scale exceeds levels that are currently sustainable on Earth. In this figure, we can show that the green is the safe operating space for each driver of global change. And you can hopefully see that biodiversity loss greatly exceeds those bounds, surpassing estimates for climate change and changes to the global nitrogen cycle. So this is how an ecosystem ecologist might think about how biodiversity loss is affecting our planet in terms of its sustainability and global systems. Alternatively, a community ecologist might think of the effect of plant diversity loss in terms such as these. The gain in productivity benefits from increasing species from 1 to 16 significantly outstrips the benefits of adding nitrogen, water, alleviating drought stress, or elevated CO2, for instance. And regardless of what perspective you come at this problem from, it's clear that the overarching theme is pretty universally accepted. The benefits of maintaining diversity, especially on plant productivity, exceed other drivers of global change. And as a result, the loss of biodiversity is a huge issue currently facing our planet, one that has implications and interacts with other drivers of global change, such as climate change, which are the two themes and the two disciplines I hope to try to connect for you today, and will continue to try to connect through my future work. So to start, let's think about this plant productivity and diversity relationship, thinking from a community ecology framework. So up here, I have a picture of Cedar Creek, which is in Minnesota. And this is one of the longest running biodiversity manipulation experiments. And most of the information we have regarding the benefits of plant diversity come from a handful of long-term research experiments, such as this one. There's another experiment like this in Germany that has been running for approximately the same amount of time. And then there's a huge number of short-term research that's been done on this as well. And even though we have overwhelming evidence that overwhelming evidence from those and other short-term experiments of this positive and saturating relationship between species richness and plant productivity, when and why these relationships are important remains less well understood. So altered precipitation and changes in plant community composition may influence when this biodiversity productivity relationship generates the greatest amount of over-yielding or the greatest productivity benefits, both of which are being altered by human influences. And this knowledge gap hinders our ability to predict where and when biodiversity and productivity relationships are the strongest and where they're most important to protect, which is what I will explore in this talk. And we'll do so by first, through an experiment that's being done at the KU field station currently, confirming this relationship between biodiversity and productivity and asking how that relationship changes with altered precipitation, as well as changes in plant community composition. And first, I have to go through and explain to you how to characterize this biodiversity productivity relationship. Okay, so they tend to characterize the effects of biodiversity through this metric called this biodiversity effect, which you can also call the net effect or relative yield. And they represent the observed yield minus what you're expecting the yield to be. And expectations are typically calculated based on how species perform in monoculture and then estimating what you'd expect them to perform at a mixture. The difference between those two is the biodiversity effect. And we partition the variation in the biodiversity effects into two potential drivers of that relationship. These were defined in the early 2000s after some very controversial debates over the validity of these experiments. And they resulted in these two different types of ways that we can generate this biodiversity effect, and they're called selection and complementarity. Selection, which mathematically is when species with higher than average monoculture yields dominate in mixture. So it will look somewhat like this in terms of a plant species. If you pretend, for instance, that all of these plants in monocultures have, for instance, 3 plants, one of these is greatly determining the amount of yield in the system. So it's when relatively few species generate biomass gains. And it's called selection because it's defined as a mechanism originating from selective processes when a relatively strong competitor is able to outperform its neighbors. Complementarity, by contrast, is when species yields on average are higher than you would expect based on these weighted monoculture means. So to translate what that means, it's when many species are driving these productivity gains from diversity. All species are doing better than we would expect. And this process is called complementarity because it evokes 2 mechanisms that imply species are able to coexist by avoiding competitive interactions. So they're complementing each other by specializing either in differing limiting resources, and that's called resource partitioning, or facilitating the development of favorable conditions. Most often, resource partitioning is the predominant driver used to explain complementarity, and I'll return to that mechanism in the second part of this talk. And we've been so interested in quantifying and describing complementarity in part because in these experiments over time, we've found that complementarity increases while selection becomes less likely as these experiments go on for longer amounts of time. So in this figure, selection is in these filled circles, while complementarity is in the open circles. And this represents the slope of the relationship between selection and richness. And we find that complementarity increases with time. So what that might look like is that there's this positive slope for both selection and complementarity in year 1, and in year 5, you still have this positive slope between selection and between complementarity and richness, whereas the slope between selection and richness is negative. And over time, with this complementarity increasing, you also see an increase in this biodiversity effect, and here they're calling it the net effect. This kind of together leads us to believe that complementarity is the primary driver of these productivity benefits from diversity over time. However, again, these have been demonstrated over long periods of time in many short-term experiments, but we have yet to explore the biotic and abiotic gradients we might expect these conditions to persist, which is what I'm attempting to address in this experiment by asking what the effects of plant richness, plant community composition, and precipitation are on these productivity and diversity relationships. And to do that, we created a grassland biodiversity experiment by manipulating plant richness to 1, 2, and 3 species mixtures. We selected from 18 species total, 6 from 3 different families. And those families, composites, grasses, and legumes, are commonly represented in tall grass prairies and also represent a diversity of resource acquisition strategies or traits. For instance, legumes associate with nitrogen fixers, and grasses have C4 metabolism that allows them to grow and metabolize at different rates than C3 plants. And we also manipulated community composition. And just as a quick note, we also overrepresented these single species mixtures to be able to do those calculations before, and we made sure to represent all species equally in all treatments so that it's completely randomized. We created phylogenetically underdispersed communities by selecting from the 2, 3, and 6 species mixtures from the same family. So they're composed of all composites, all legumes, or all grasses. And then for comparison, we also created phylogenetically overdispersed communities by selecting species from differing families. As for instance, having legumes and grasses together, and then in these larger families, having all species together. And I'll try my best as we move throughout the talk to refer to these as one family or single family mixtures and multiple family mixtures. So it's a little bit less jargon heavy. And lastly, we manipulated precipitation using these large rainfall manipulation shelters at the KU field station and irrigating to 50 and 150% of annual local rainfall. Okay, so to quantify plant productivity, we used multiple different measures of productivity. First, I'm actually going to go through this bottom one. We harvested biomass from 0.1 meter squared strips, sorted them to species, and dried them at 70 degrees C and weighed them. And we did that sampling from 2019 until 2020. So in year 2 and year 3, we did not harvest biomass in year 1 to allow for establishment. However, we did assess overall plant cover by looking at the total amount of plants that are growing over the entire plot. And we did that at peak biomass from 2018 until 2020. So for this first part of the talk, I will present those data because we have the most complete data set. And then in the second part of the talk, I'll present data that looks at how this cover changes temporally throughout the season. So I also went out and looked at the planted species and their cover from May until October to see how the cover of these plants changes throughout the season. But again, this first part, I'm going to focus on this peak biomass data that's collected from cover. And the reason I'm doing that is because we have the most complete data set for that. And then again, I will partition the variation in these effects into complementarity and selection, which is when many or few species are deriving these productivity gains from richness. And I'll present them as a regression line with confidence intervals and the effect line of 0. So when these confidence intervals are not overlapping with 0, that effect is significantly greater than 0. And when it's above 0, that's a positive effect. And when it's below 0, it's negative for that particular metric. Okay, so just to get into that data, the first thing we would like to do is confirm whether or not we have this productivity diversity relationship. So we ask, what are the effects of plant richness, very simply, on plant productivity? And I'll look at that for every single one of those productivity measurements that I described earlier. So selection, complementarity, then I call them the biodiversity effect, relative yield in this case. And then ultimately, I'll also present these yields related to richness. And what we see is actually quite simple and quite consistent. Richness positively predicts all measures of productivity. We had early establishment of this positive relationship between productivity and richness in 2018, which was really exciting to see and actually quite surprising. Though the effects were not quite significantly greater than 0, that did not occur until 2019. And then in 2020, we saw the same trends or similar trends to what we saw in 2019. But what was notable is we started to see a little bit more variation in specifically selection and complementarity that year. So overall, being able to establish this pattern was really, really exciting to see. And we do see that plant richness consistently increases all of the different types of plant productivity measurements that we had in our plots. However, there's still a great deal of variation in our data that remains unexplained. So we specifically looked at whether or not community composition and precipitation can explain some of that variation in selection and complementarity. So I'm going to start going through the effects of selection. And again, selection is when few species drive these productivity gains, and what they might look like in a single and multiple species mixture is kind of depicted here. And what we observed in 2018 and 2019 was a significant positive relationship between selection and richness, which I described later. And notably, there was really no difference in the steepness of slope between single and multiple family mixtures, so this gray and this purple line. Except for legumes, we do see significantly steeper slope and legumes for that relationship, specifically in 2019. So we had significantly greater selection in that legume family in the first 2 years. In 2020, interestingly enough, we observed this negative relationship between selection and richness in composites, which are these little flowering plants like echinacea that I describe up here. And the differences between single and multiple families start to become apparent. So selection in grasses caught up to legumes, which suggests that selection we observed in those previous 2 years by legumes is likely just because they got out faster, but not because they have better or they're more likely to have higher selection than other families, which was really encouraging to see. And then there was also a significant interaction between community composition and precipitation. So here I've been presenting the relationship for the 50% water treatment. And what we see in these higher water treatments is that there's greater differentiation between these single and multiple families. So the slope of the relationship between selection and single families is steeper, and overall selection is higher in these single family mixtures and is somewhat comparable to what you see in these 50% precipitation treatments. However, in these multiple family mixtures, selection is significantly lower. It overlaps with 0 at some mixtures, and it increases less with richness and is lower overall than what you would see in these 50% treatments. And that may be a little bit overwhelming to think about, but when you aggregate these two trends together and think about them, and if we exclude that negative relationship between composites and this 50% water treatment, there's a significant difference between selection by water treatment overall. So in the 50% water treatment, we're more likely to see higher selection than in these 150% water treatments. So just to summarize, we are more likely to see higher selection in lower precipitation. And in higher precipitation, we're more likely to see high selection when families are composed of a single family, or from a single, when mixtures are composed of a single family. Excuse me, let me say that one more time just to make sure it's really clear. Okay, so in these low precipitation treatments, we see high selection independent of community composition. And in higher precipitation, we see higher selection when mixtures or plots are composed of plants from the same family as opposed from multiple families. Hopefully that's kind of clear, but just overall, the take-home point is that we get higher selection in water-limited systems. Okay, so now let's explore these relationships with complementarity, which is when many species drive these productivity gains from richness. And what we see in 2019 and 2020 is a very simple positive relationship between richness and complementarity. There are no effects of community composition or precipitation in that year, in either of those years. So it's just very simply complementarity is driven by increased richness. In 2020, we start to see the effect of precipitation on complementarity. We see that complementarity is significantly higher in these 150% water treatments in comparison to these 50% water treatments, but there's no difference between the slope of that relationship. So overall, we still see an increase in complementarity with richness that occurs at the same rate between those two treatments. They're just shifted up in higher precipitation. So that suggests that many species are likely driving the productivity benefits from diversity in higher precipitation systems. So just to quickly summarize, we do see that diversity matters overall, but this interaction between precipitation and plant community compositions determine when few or many species are likely to drive those productivity gains. In lower precipitation, few species are likely driving those gains from those productivity gains from richness, and in higher precipitation, many species are driving those patterns. And so how do those patterns aggregate up to these biodiversity effects, to these net effects, and how do they actually affect overall yields? Well, really quickly, just to show you the simplified pattern we have with precipitation and selection and complementarity, you can see that selection is significantly higher in low rainfall, while complementarity is significantly higher in higher rainfall. And since the relative yields are selection plus complementarity, the effective rainfall is marginal on relative yields. So you have slightly higher relative yields in these higher precipitation treatments. But when you scale that up to cover and total yields, you actually don't see a difference in yields between these high and low precipitation treatments, which is really interesting. So when you link these biotic and abiotic conditions, we saw that lower precipitation, relatively few species drive the productivity benefits from diversity. So we saw higher selection in water limited systems. And this likely has implications for the stability and resilience of those systems. We might expect a water limited system to be more susceptible to losses of productivity as a result of a random event or an extreme event. So ecosystems that tend to depend on few or single species for critical functions like plant productivity are vulnerable to disturbances. If, for instance, a disease or a really good invasive species happened upon that system, we might expect that they are at greater risk of tipping into an alternative stable state that might not be able to maintain that same level of productivity that we saw. However, even though selection can drive productivity benefits in water limited systems where we might not otherwise expect it, it it does show that even across precipitation gradients, we still are able to maintain this diversity productivity relationship. And that finding supports and confirms what's been demonstrated in many and many other biodiversity manipulations experiments that, productivity does increase with plant richness. And by contrast, in higher precipitation, complementarity drives from the productivity benefits of so by contrast, in higher precipitation, complementarity drives the productivity benefits of diversity. And that's independent of precipitation and plant community composition, which is actually really encouraging and kind of fascinating to see when complementarity can increase independent of whether or not communities are assembled from many or one family. That just adds evidence that these findings and these patterns are robust and likely not a function of the communities that we're selecting by selecting random species from these lists. So again, this supports what has been shown time and time again over long-term biodiversity manipulation experiments. And so I know that some of those interactions are quite complicated and there are some interesting interactions that I have explained, but the take-home message is actually quite simple. It's that diversity matters. It does generate productivity, and it can do so across both biotic and abiotic gradients. And then finally, that the loss of species can adversely affect productivity of these systems. All of which answers when we expect to see when diversity matters. However, we have yet to address why complementarity increases with richness. Earlier, I alluded to complementarity being driven by resource partitioning, which occurs when a species can avoid detrimental competition by specializing in differing resources. However, while resource partitioning is implied from that term, it is important to actually test how well it explains the patterns that we are seeing, which is what we'll do in this part of the talk. So just really quickly to break out how resource partitioning works in low and high diversity systems. In low diversity plots, you might expect fewer species, and as a result, fewer different resource acquisition strategies. That will result in greater competition between species of the same population for that same limiting resource, which adversely affects growth. So they'll be allocating more resources to competing with their neighbors as opposed to growing. In comparison, in high diversity plots, where there are more diverse resource acquisition strategies that may enable these plants to, one, not have to compete with their neighbors for the same limiting resources, and two, to be able to allocate more of the resources that they're gaining from the system to growing larger, which is the theory of how we generate over-yielding, more yielding than we'd expect based on these monocultures. Most studies address resource partitioning by assessing the diversity of resource acquisition strategies within the plant community. So for instance, it might look something like this, where you have a bunch of different plants, some of which fix nitrogen by associating with by forming symbiotic relationships with microbes that can fix nitrogen, some by employing different metabolic rates that allow them to grow faster, some by growing taller, and some, for instance, by rooting to different depths or laterally in different ways, all of which tend to be measured and incorporated into these resource acquisition strategies, or into a metric that describes resource acquisition strategies. But we additionally wanted to take that one step further and explore if differences in how plants grow over time can also explain resource partitioning, because growing at a different time of year is another way to more thoroughly exploit these resources at different time points throughout the season. So just quickly, we are addressing, do we find evidence that resource partitioning is why we see the strong productivity and diversity relationships? And to what extent is that driven by diverse resource acquisition or diverse temporal growth strategies? To do that, I quantified resource acquisition strategies in a way that's really commonly used in these experiments and is relative to the growth of the plants in those plots. And I don't necessarily want you to focus on what traits I use to quantify it, but just know that when that value is low, you have fewer differences in resource acquisition strategies. So this trait diversity is low. That means there's less resource acquisition strategies within that plot. And when this value is high, it means there's more differing types of resource acquisition strategies. And then I assessed cover over the season, and I analyzed it by looking at the variance in the total cover in the plot and comparing that to single species. And when I take that metric and when it's low, so closer to 0, because I take 1 minus that metric, it means that all of those plants are growing at relatively the same time. So when this value is low, it means that plants grow at the same time point throughout the season, whereas when it's high, they are more likely to be differentiating in their growth or their phenology throughout the season. And just again, to reiterate, we're trying to see whether or not this predicts complementarity, which is the common, because resource, So we're trying to see whether or not that predicts complementarity, because resource partitioning is the mechanism invoked to describe complementarity overall. Okay, I'm going to start with these temporal diversity metrics, so these plant phenologies. And we actually do see a positive relationship between this community temporal growth strategy, or asynchrony as I call it down here, but these different temporal growth strategies and complementarity in 2019 and 2020. And just really quickly, I didn't do these temporal cover measures in 2018, so I don't have data, which is why I'm only presenting it for 2019 and 2020. We saw no relationship between selection and increased differences in temporal growth strategies. And the result on the net effects, or this relative yield, or this biodiversity effect, was positive. So we do see evidence that differing plant phenologies contributes to productivity benefits from diversity, and it does actually predict complementarity. So let's look at these resource acquisition strategies as well, which is the one that's actually more often used to describe complementarity. And again, as we expect in both years, we see 2019 and 2020, we see complementarity increases with these functional traits. So in plots with higher functional trait diversity, we get higher complementarity. However, we also observed a negative relationship between the selection effect and these functional trait diversity metrics, such that we actually have no detectable relationship between functional trait diversity and this relative yield metric. And notably, the relationship that we saw between selection and this trait diversity effect has been persistent every single year of the study. So though this pattern was somewhat unexpected, or maybe it could have been expected, but we didn't necessarily predict it coming forward, its continued presence in our data kind of suggests that it's not an anomaly, and it's something that we'll have to think about later on. So then when we consider both these mechanisms together and scale them up to their effects on overall yields, which I'm presenting as total cover, what we actually see is that because of those net differences in the way selection and complementarity drive this functional trait diversity, we do not see a significant impact of functional trait diversity on overall yields. But we do see a positive relationship between differing plant phenologies or differing temporal growth strategies and cover both years. However, notably, those relationships are somewhat weak, explaining very little of the variation in the data overall. And that's actually true for complementarity as well. We're explaining less than 10% of the variation in the data with these two differing metrics of resource partitioning. So while resource partitioning is implied by the term complementarity, and it's really important to actually test how well it explains these productivity benefits that we're seeing. And in doing so, we are able to confirm that resource partitioning does, or as it's measured by functional traits, does predict complementarity, and differing phenologies also predict cover, right? However, they explain much less of the variation of the data than we might expect if resource partitioning alone was the sole or only driver of complementarity. And we think that suggests that alternative mechanisms may likewise be contributing to these patterns. So I mentioned before that facilitation is also evoked with resource partitioning as a potential driver of complementarity. And it occurs when plant communities facilitate the development of favorable conditions for other species. However, it's really hard to directly measure facilitation, especially at a single time point. But alternatively, these patterns can arise from pathogen dynamics in the system as well. And that's a hypothesis that's really easy to test, and one that my collaborators are currently tackling. So these diversity benefits as resulting from pathogens actually kind of are generated in a similar way to these resource partitioning pathways. And instead of exerting energy to combat your neighbor, you're exerting energy to combat pathogens or natural enemies. So when you have more specialized pathogens in the system, the plants are not able to grow as big because they have to allocate more resources to defense. In contrast, in these high diversity systems, when you have maybe a greater diversity of pathogens, but fewer specialized pathogens, they're diverting less energy to defense against these specialized pathogens and more to growth. And that's a very testable hypothesis that can help hopefully explain some of the variation in these data that we've left unexplained. So just to reiterate, because I know that was a lot, right? But the take-home messages, I think, are quite simple. We are able to confirm this biodiversity and productivity relationship. And though there are complicated interaction regarding how that relationship arises, overall, we do show that diversity matters. It generates productivity, and it does so across both biotic and abiotic gradients. And then though complementarity is explaining some of the variation, or is explained somewhat by resource partitioning, it's not explained quite as much as we would expect if it's the sole driver of these patterns, suggesting that other mechanisms may be working in consort with resource partitioning to generate these productivity benefits from richness. Okay, so up until this point, we have dug into how plant diversity stimulates plant productivity. And we've looked at this pattern and mechanism. And for the most part, we've been using this ecosystem or this community ecology framework. We've been thinking about how plant diversity drives plant productivity. So what are the implications of these patterns for other measures of ecosystem function, right? So from the perspective of an ecosystem ecologist, we might and do wonder how these patterns influence other processes like nutrient retention or carbon storage. We know carbon storage, which is of particular interest given the current trajectories for global CO2 emissions, is a function of carbon taken in by a system and the carbon lost from the system. And ultimately, the implications of productivity benefits from diversity on carbon storage are dependent on the respiratory losses from soils, which can be influenced by both above and below ground communities, as well as these abiotic conditions. So to step back and think about soil respiration on a whole, right? There are well-defined relationships between abiotic drivers and soil respiration. But on a global scale, these abiotic drivers alone, specifically soil moisture and soil temperature, fail to predict respiration when you scale up. And difficulties incorporating the complexity of these biological communities and their sensitivities to changes in precipitation are suspected to account for those difficulties. And it remains an unaddressed challenge that needs to be tackled to scale predictions of respiration globally, which we are able to address with our current experimental design. So thinking about how plant communities can influence soil respiration, above ground productivity does positively predict soil respiration in some instances. And plant diversity, as well as plant community composition, can likewise increase respiration from soils. However, these patterns likely correlate with changes in root respiration or changes in below ground microbial activity. So ultimately, soil respiration is driven by the respiration of roots or the respiration of microbes. And root respiration, and then above ground communities can mediate root respiration by simply producing less root biomass or greater root biomass, right? And then alternatively, above ground communities can mediate microbial respiration in soils through a variety of pathways. Here I'll note two. One, they can form symbioses with microbial communities. Here I'm depicting nitrogen fixers and arbuscular mycorrhizal fungi, which form symbiotic relationships with plants commonly in these tall grass prairie systems. And by doing so, the plants are able to give these microbes carbon, enabling them to potentially produce more biomass, which can increase microbial respiration in the system. Alternatively, they can stimulate microbial respiration by providing substrates for the rhizosphere communities as a whole. So just the communities living generally in soils and around all of the roots, not necessarily associating directly with the roots. And what's nice about our study system is that we are able to address those mechanisms and also these linkages between the abiotic and biotic condition with our current study design. So in this part of the talk, I'll ask if species richness, plant community composition, and precipitation alter CO2 respired from soils. And then I'll also ask what mechanism, root, or microbial respiration is driving these losses of carbon from soils. Qualitatively at this point, though, just as a foreshadowing, we do have the data to eventually be able to look at those more quantitative as well, from a more quantitative perspective as well. And to do this, we measure soil respiration from in situ collars. We did this in 240 plots monthly from May until October in 2020. We took congruent measurements of soil moisture and plant cover and then calculated growing season averages of all of those to look at the effects of our treatments alone and to minimize some of the variation that you might expect in those naturally to occur over the season. And then I will model growing season average soil respiration looking at our treatments, all of the interactions thereof. And then we also accounted for the common covariates, specifically plant cover. And then I incorporated moisture and as well as a moisture squared term because moisture and soil respiration are well documented to have a plateauing relationship with respiration. So it increases as water limitation is alleviated, flattens out, and then at some point water becomes too abundant and actually limits respiration. So that's why I have both of those in this model. And really quickly to show you how the covariates or how these pan out when we actually put them in the model, we see a significant block effect. We do see a marginal effect of this moisture squared term in our model. And though cover and moisture alone are not significant parameters in our model overall, their inclusion significantly improved fit, so we left them in as covariates. But then to look at our actual treatments, we saw a marginal positive effect of richness on respiration, note the log scale. We saw significantly greater respiration in these multiple families mixtures and compared to the single family of mixtures. And we saw significantly higher respiration in higher precipitation, though there's a significant or marginal two-way interaction between every single one of our treatments. So we have to consider them all together. And when we do so, what we see is that there's an increase in the carbon dioxide lost from soils in high diversity plots, especially those that are formed from multiple families. And that relationship strengthens with precipitation. So we see greater CO2 loss from high diversity plots that are formed from multiple families. We highlight again these single family mixtures, we see a great deal of variation that remains unexplained. And when you partition that out into which families are actually driving these trends, what we actually see is quite interesting. We see that both grasses and multiple species mixtures account for this increase in CO2 loss from soils in these high diversity plots. So it's not just multiple family mixtures, it's also these single family grass mixtures that are increasing soil respiration in high diversity plots and strengthening that relationship with higher precipitation. While conversely, legumes and composites account for a decrease in soil respiration with richness in these low precipitation plots, and then no relationship in high precipitation. I know that might look confusing, but remember this is a log scale. So this is actually a decline. This is no relationship. And we suspect that the fact that grasses are increasing respiration with richness might suggest that a greater biomass might be driving some of these trends. CU4 grasses are really well known to have high density of fine roots, which are known to respire quite a bit. And though we don't quite, and in comparison, right, these like low, and in comparison, the legumes, which tend to form symbiotic relationships with nitrogen fixtures, don't seem to be increasing soil respiration. So it kind of hints at this idea that these root biomass and these root dynamics might be driving this, which we'll be able to test through soil samples that we collect and that we're processing currently for root biomass. So just to really quickly summarize, the increase with increasing richness, we see greater carbon dioxide loss from soils. With plant communities consisting of multiple families or grasses, and importantly, that relationship strengthens with precipitation. And so we might suspect, you know, the lowest amount of carbon dioxide loss from species that look across these gradients. Like, for instance, here we have all legumes, and even regardless of diversity, we expect less CO2 loss from those systems, especially in lower precipitation, whereas we expect the most loss from these multiple family mixtures and from grass mixtures with higher precipitation. And then how does that scale to carbon storage, right? Which is what I was talking about earlier on. And to think about that, you actually have to look at the biomass. So this is the first time I'm presenting biomass data here instead of cover as yields. And it's in part because we have that data to be able to pair with our 2020 soil respiration. And what I want you to note is that the species that account for the greatest or the steepest increase in biomass with richness are actually composites and legumes. So in those plots, we have the greatest amount of carbon that is being brought into those systems. But conversely, we also seem to have the least amount of soil respiration in those systems, which does kind of suggest that we might actually see the greatest amount of carbon stored in those systems in comparison to, for instance, grass systems. And future directions are to address specifically that, and also to address some of those mechanisms a little bit more clearly. So for year 3, we also collected we collected soils to quantify microbial community composition, which we're currently processing, and we hope to be able to link those two. But I also collected soil samples and did a bunch of nutrient analysis on them, as well as quantified the microbial biomass, and we're currently looking at root biomass to be able to more directly link those mechanisms. I likewise did a leaf litter decomposition story or experiment, which can explain some of the carbon that's being put back into soils from the decay of these plant litter. And then I also did a root inclusion exclusion experiment so that we can look at how roots might actually be stimulating these microbial communities in soils. So overall, we have a lot of data to fill out this carbon story that we haven't, which we can kind of allude to at this point, but we actually don't have the full data set for. So just to quickly summarize, because that's a lot and I know, where we've been in this last hour. So overall, we were able to confirm this biodiversity and productivity relationship. And though there were complicated interactions regarding how it arises across these gradients, overall, we do see that plant diversity does generate plant productivity across both of those gradients, abiotic and biotic gradients, which was really encouraging to see. And resource partitioning does explain complementarity to some extent, however, not as much as we would expect if it really was the sole driver, suggesting that other mechanisms are likely working in consort with resource partitioning to generate complementarity in these systems. And finally, those were our community ecology hats that we put on when we put on our ecosystem ecology hats. What are the implications for these patterns on other measures of ecosystem function? And here we explored carbon loss from soil respiration and found in plots where we have observed the greatest increase in biomass with richness, we've also observed the least amount of carbon losses from soil restoration, suggesting though the interactions between abiotic and biotic communities may be able to generate comparable levels of productivity across these diversity gradients, they may not result in the same level of carbon storage as in the soil, though. Just the quick note is that we do have to do more work to confirm this, but it's just an interesting nuance for how these patterns might actually have implications for overall carbon budgets on a global scale. And then moving forward, where we're going from here, I just put up this figure from a very, very, very recent publication in Nature Communications. It's called Blind Spots in Global Soil Biodiversity and Ecosystem Function Research. And here they characterize the geographic extent of all of these global soil biodiversity and ecosystem function studies in terms of whether or not they quantified biodiversity, biomass, or ecosystem function. And what I really quickly want you to focus on is right over here. Here you can see that they characterize what kind of soil biodiversity is being quantified and what kind of ecosystem functions are being quantified. And then they looked at what percentage of these studies overlap, right? And here in this 0.01%, you see that there's this intersection between biodiversity, biomass, and function. And I think what's really exciting is there's the least amount of research on those topics, but it's one of the ones that's of most interest to this literature as a whole. And I think we're really uniquely situated in this experiment to further explore that. We've measured biodiversity in the soil communities. We've measured almost all of these ecosystem functions in these plots. And we also have biomass measurements for all of these. But this is just for soils, right? And I think what's really cool to think about is that we also have these data for plant communities as well, which is a huge blind spot in this research as well that I think will be really interesting to continue to explore. So one of the coolest parts about this study and moving forward, what I hope to think a lot more about is how to link these community ecologies and ecosystem ecology disciplines and thinking about biodiversity and ecosystem function. And I'm really excited about one, being able to contribute to this very, very small and emergent and important body of literature, but also being able to take it one step further and link these plant communities in with them. And with that, I would like to thank many of people for help with this research, my committee, the collaborators, the many technicians associated with this project, to these people who helped me understand the community ecology of this biodiversity ecosystem function research, and to my funding sources and our data sources as well.